I'll meet you at the mall. I was like, so we
Like, the thing is that
somehow I get him out there.
I kind of know what's going on, but I just can't get the right answer. But you gotta just move the computer over this.
I think it's fun. Great.
All right, testing. Okay. Where's the sign-in sheet? Add the sign-in sheet. Here's the sign-in sheet. All right. Let's see. This is good. Good. All right. So, continuing on from last time. Last time we talked about the central dogma of molecular biology. Where is it? Here it is. The central dogma of molecular biology, where the three most important components to life, DNA, RNA, and proteins. DNA has the capability to self-replicate, but in order to actually have some functional relevance, DNA by itself doesn't do anything. DNA undergoes this process of transcription to become mRNA, and mRNA undergoes a process of translation to become proteins. Proteins are your functional component of life. You got structural proteins, enzymatic proteins, and other ones that contribute to various processes in the body. So from this point on, you guys learn about all these mechanisms essential for life. We talked about the different nitrogenous bases. There's two categories, purines and pyrimidines, and you are supposed to know which nitrogenous bases belong to which category. You don't have to know the special RNA bases like these. These are very rare bases. We talked about the different properties of nitrogenous bases, and I think we got all the way to nucleosides. So the last thing we talked about last time was nucleoside. So what a nucleoside is, emphasis on the S, nucleoside, is a nitrogenous base attached to a sugar. Last time we talked about how to name nucleosides. The rule is listed right here. It is very simple. You just change the suffix of a particular nitrogenous base to describe its nucleoside, and we practiced identifying nucleosides here. And the very last thing, and this is where I'll start here, is this interesting fact. The very last thing of last time, and then we'll start here, is the fact that of all of your nucleosides, it turns out that adenosine is the only one that actually does anything. Meaning adenosine just by itself is the only one with bioactivity. All these other things, cytidine, uridine, guanosine, and thymidine, are unable to do anything unless there's phosphates attached to them or some other modification attached to them. Adenosine by itself is able to, and it's listed here, it's able to act as a vasodilator, muscle relaxant, and produce sleepiness. Interestingly enough, caffeine blocks the bioactivity of adenosine. And I always tell this interesting fact here. How many of you have heard of something called a caffeine nap, or like a coffee nap? Has anyone heard of that before? And then nap for like 20 minutes. All right, so here's what a caffeine nap is. It's an interesting little thing, and it's based on this activity here. A caffeine nap, supposedly how it works, is that you drink a cup of coffee within like a short amount of time, like you try to drink a whole cup really quickly, and then you actually take a nap for like 20-25 minutes. And then when you wake up, the caffeine actually has an even stronger effect on you, because it takes about 20-25 minutes for the caffeine to really, for the coffee to get digested and the caffeine to get processed, and then it actually does something. So the nap, it turns out, supposedly, the nap clears out the receptors for adenosine, and the caffeine blocks those receptors for adenosine. It's an interesting little fact. You won't be asked about it on the exam, but if you ever go Google something called like a caffeine nap or coffee nap, it actually supposedly has some biological relevance. Again, the nap clears out the receptors for adenosine, and the caffeine actually blocks more adenosine from attaching to those receptors. That's why you feel extra awake after that 20-minute caffeine nap. So you can try that if you're studying for biochem. Moving on, stuff that may be asked about. So contrasting to exam 1 and 2, where you didn't really need to know really specific biological activities of sugars and lipids. You kind of just need to know very general stuff. Here is one of the weird parts of the course where you actually kind of need to know interesting facts and uses of some of these things here. So the very first thing we talk about is a use for adenosine, or a role for adenosine, and it's listed here. Vasodilator, muscle relaxant, and produces sleepiness. The very next thing is a compound called azacitidine or deoxyazacitidine. Don't worry about memorizing the structure of this thing, but if you see its name, make sure you know the following about it. Let me find, I wonder if I should talk about something else first. Let me think real quick. Okay. Yeah, I'll talk about this thing first, and then I'll explain what azacitidine does. So, can I zoom out here? Is that as far out as it goes? Okay. So, a little bit of intro to transcription and gene expression stuff. So we know central dogma tells us DNA gets transcribed into mRNA, gets translated into proteins. Okay? Supposedly, and you may hear this in other classes, there are some genes called proto-oncogenes. Has anyone ever heard of that term before? All right, for those who haven't, I'll explain what these are. So some genes, it actually turns out, are bad in that if they are transcribed and then translated, it actually causes cancer. So they call those proto-oncogenes. And when a proto-oncogene has one slight alteration to it, it becomes an oncogene, a gene that causes cancer. So what is happening at the DNA level? So I'm going to draw a strand of DNA, and I'm going to illustrate right here that there's a gene on it. So I know we haven't talked about DNA replication or anything yet, so maybe visualizing this may not be as clear as it can be right now, but I'm going to try to illustrate something here. So DNA, lots of bases. There's a region of DNA that represents a gene. I'll just call it gene A. Okay? In order for the protein to be made, you have to transcribe this gene. So there's a whole bunch of enzymes that come in. You don't have to know the enzymes yet. We're going to get to those eventually. A whole bunch of enzymes come in and they start reading the DNA strand. And they actually read this gene and they produce an mRNA strand. And I'm going to call this mRNA strand for gene A. Okay, so in transcription, you make this thing. Okay, and then eventually this mRNA strand is going to go to a ribosome. And the ribosome is going to translate this thing into a series of amino acids. And this is going to be the protein for gene A. Seems simple enough. There's a lot of like things going on in between each of these steps that affects this process. For example, and I may get this backwards, so we'll fix it later. There are certain ways you can modify your DNA without even causing a mutation. You can modify your DNA in a way that it will affect the transcription and translation of a particular gene without technically being a mutation. One of those ways is a thing called DNA methylation, which is in parts, DNA methylation is a natural thing that happens to everyone over the course of your lifespan. What DNA methylation is simply as you grow older and due to certain events in your life, methyl groups start accumulating at various spots in your DNA. They're not mutations because they're not technically changing anything with the bases in DNA. Like again, I didn't draw them in, but imagine that A's, T's, G's, and C's are all written in here. It isn't a mutation, but this methylation of DNA is something called epigenetic modification. Or an epigenetic change, whatever word you want to use. The key phrase in this statement is epigenetics. Epigenetics is this pattern of methylation that naturally occurs as you age and due to certain like life events and lifestyles. But methylation starts accumulating. Turns out sometimes some genetic diseases occur due to an accumulation of methyl groups at a particular gene. And again, I might get this backwards, so when we go back to the PowerPoint, I'll give you what that particular disease was. This is still true here, it just might not apply to the disease that I'm going to talk about later. And methylation accumulates. What ends up happening is that if enough methyl groups accumulate on a particular gene, you lose the ability to transcribe it. You didn't mutate the gene at all. As far as your enzymes would know, your gene is normal. There's nothing to repair here. But because all these methyl groups started accumulating on it, enzymes don't understand how to transcribe something with so much methyl groups on it. So enzymes will just skip over it, and this gene you end up losing the ability to transcribe this gene. And if you can't transcribe this gene, you very clearly can't make proteins representing this gene. So the expression, which is the level of this gene, the level of transcription and translation of this gene ends up going down simply because of methyl group accumulation. Some diseases, such as acute myelogenous leukemia, occur because of this pattern of methylation, or in some other cases, acetylation, that affect gene expression. So I'll start with methylation because I think it's kind of the easier thing to illustrate. I can't remember if it applies to that disease, but methylation is a natural process that occurs where you block the expression of a particular gene simply because the methyl groups get in the way and you are unable to transcribe that gene. Okay, so I'll give you that little bit of an intro, and now we'll actually see how this disease particularly works. Let's see if I had it the right way or if I said it the opposite. Some disease. So there's a disease, and you can read this in detail later. There's a disease called acute myelogenous leukemia. I wish I could find where it says what it does. I kind of get it backwards. Let me see real quick. I don't care. That's not it either. Oh, okay, yeah, so I was right. Phew, okay. So this disease, acute myelogenous leukemia, actually occurs because of methylation of a particular gene. And in doing so, for whatever reason that you don't have to know specifically for this disease, but the methylation of a particular gene causes the condition known as acute myelogenous leukemia, which is a very fast-acting, really serious blood cancer disease. For the longest time, they had no idea how to treat this because as far as they could tell, there was nothing wrong, there weren't any mutations or anything. And then they found eventually that it was caused due to a methylation pattern on a particular gene. So, due to some research that was going on, someone found out that this drug here, or this compound called azacitidine, which is a nucleoside analog, now we finally get to the reason why he's talking about it here. It is a nucleoside analog, so we just talked about nucleosides. This nucleoside analog, it turns out, actually shuts down the enzymes that cause methylation on genes. So if you read this statement right here, azacitidine causes an inhibition of enzymes called DNA methyltransferases. So these enzymes normally go around and for reasons we don't understand yet, attach methyl groups to genes as time goes on. So this drug inhibits these enzymes. Therefore, if I go back to the picture here, if it inhibits the enzymes that are methylating DNA, there's going to be less methyl groups on them, which is good. That means that whatever this gene was can continue being transcribed and translated as normal. So this drug is what is called an epigenetic drug, because it is affecting the epigenetics of your genome. It doesn't do anything with the actual bases of your DNA, it just affects the methyl groups on DNA. All right, so it's an epigenetic drug. There's some statistics and stuff on here. The bottom line is this drug turned out to be so effective that, I don't remember what the number is, but the prognosis for people with acute myelogenous leukemia instantly improved dramatically as long as they took this drug. And again, all it does is it shuts down the enzyme that methylates DNA. Therefore, this disease, which was caused by overmethylation, was able to be treated. There's a couple extra interesting facts on it, but I do know in terms of what he usually asks about azacitidine, it boils down to that, is that it's an epigenetic drug and is used to treat myelogenous leukemia, and that azacitidine is a nucleoside analog. And we take a look at it. It kind of looks like a nucleoside, right? Here's a nitrogenous base and here's a ribose sugar down here. I'm not going to look too closely at what the difference is between this and regular cytidine, because you won't be asked anything about this structure. But as long as you associate azacitidine with nucleoside analog, you'll be fine. And its biological use here, which is listed in these slides. All right, so that's one interesting fact. We'll move on to some other interesting facts. So here's some more facts about DNA methylation in general. I talked about the specific example regarding acute myelogenous leukemia, but it turns out that DNA methylation, especially if it's more than normal, is associated with mortality. And what else do we have here? And is usually correlated with poor lifestyle. Like, I don't know if it says it here exactly, but if you smoke a lot or you drink a lot, you actually, it turns out your genome, your DNA actually methylates at a faster rate than people living a healthy lifestyle. Because of that, this current research is kind of finding that there's an association between these epigenetic changes and poor, shorter lifespans and poor health outcomes later in life. So, for the longest time, we know that, obesity and all these things are associated with, poor life outcomes. Only now are we maybe figuring out why so many different things seem to associate with a poor life outcome, and this may be one of the things linking all that stuff together. It is epigenetics, DNA methylation. We'll talk about, and I don't know if it's here. Oh yeah, good timing. So why is DNA methylation and other chromatin modifications so important in terms of like normal activity? Let me see, maybe I'll draw it here. Yeah, okay. So there's a bunch of extra facts on here, and I'll go back to it later, but let me talk about chromatin in general. And again, this is weird because you technically won't learn about the details about chromatin until like chapter 28 and 29, but since he talks a little bit about DNA methylation here, I think it's important for you guys to know about chromatin. All right, so back to these drawings here. So in eukaryotes, all right, which what we are, in eukaryotes, DNA strands are wrapped around these things called histones. Okay. Wrapped around histones. So I'm going to attempt to illustrate this because I don't think there's a diagram for this in the PowerPoints. So here's a bunch of histone proteins. Histones are proteins. Okay. And in eukaryotes, because DNA is so large, DNA is so long in eukaryotes, they have to compact it close together, and they do so by wrapping strands of DNA around histones. So imagine there's many histones, and about every 200 base pairs, you have a histone and part of the DNA wrapped around it. Okay? And all that put together, all your histones and all your DNA put together is what makes up your genome. Which what makes each person, and what determines each organism from each other is how DNA is wrapped around histones and what genes are found in DNA. So, this structure here is what is chromatin. This thing here, this interplay between DNA and histones, can change due to a bunch of different reasons. We learned one reason already. DNA methylation affects chromatin. We talked about one way, right? If DNA is methylated, whether here or here or here, doesn't really matter. What did we say? What happens to transcription of a gene if DNA is methylated? Is less, right? You are unable to transcribe a gene if it's methylated, right? There's other things that you can do to chromatin to affect transcription. Another one is DNA or I believe it's also histone acetylation. Okay. So again, I know it might be hard to visualize this because technically DNA hasn't been covered and chromatin hasn't been covered yet, but it is important to understand the drugs that we're talking about here. So DNA methylation was one thing we found out. We can modify the genome. And when you methylate DNA, you end up inhibiting transcription. DNA or histone acetylation does the opposite. There are enzymes that add acetyl groups to these things. I think it's OCH3 or OOCH3, I can't remember exactly. You don't have to identify an acetyl group or anything. But imagine that there's an opposite mechanism called DNA acetylation where you attach acetyl groups to histones. This does the opposite. It turns out if DNA methylation inhibits transcription of a gene and thus decreases the expression of a gene, histone acetylation actually increases transcription of a gene. The reason why it does this is that what histone acetylation does is that when it attaches an acetyl group to these histone proteins, DNA actually ends up unwinding from these histones. And whenever DNA is unwound from something, when it's a little bit of in a looser orientation, it is much easier to transcribe. And trust me, I am oversimplifying this, and I know there's a lot of terms technically a lot of you may never have heard before, so I do suggest you re-watch the video on this, and I will talk about this in detail later. But the general idea of epigenetics boils down to how you modify chromatin. We learned one way is that by methylating it, you inhibit the transcription of genes. Another way is that by acetylating histones, you increase transcription of genes because these histones can be removed. DNA can unwind from histones and it's easier to transcribe it and express certain genes in DNA. Okay. So, this interplay again leads to some interesting things. This interplay of chromatin and the state of chromatin and how it changes, it turns out that chromatin changes as something develops and differentiates and ages, okay? So I'm going to oversimplify this, and he will probably, I don't know if he's even gotten to this yet, but if he has gotten to it, he may go into more detail than you need for the exam. But it turns out when any organism is in development, right? There's all these terms here you have to know in like a genetics or anatomy course. You don't have to know any of these terms for this class. But in development, your chromatin is in one particular orientation. It's in one particular orientation in early development, sort of illustrated here on the left, that chromatin is in some particular orientation in early development. Notice how chromatin is opening early in development. So based off our illustration we had earlier, probably a lot of DNA is unwinding from histones early in development. What that causes is that an increase in transcription of a lot of different genes necessary for an organism to develop. If you want to develop from a 1-cell thing to a 2-cell thing to a 4-cell and to a very complicated organism, you need to be transcribing a whole mess of genes. So chromatin opens up early in development. As you develop and eventually as your organs and tissues continue developing and reaching their finished form, notice chromatin closes back up. Okay, that's because you don't need to transcribe millions of things very quickly anymore because you're finishing development. And as chromatin closes back up, transcription of many genes closes down. Okay, and now once the organism or you are born and alive, you have a constant low level of transcription of only certain genes you need to maintain your bodies and your body and your cells and living. You have a low level of transcription. So that's what this is trying to tell you, is that chromatin changes during development because you have to develop the organism. You develop the organism very quickly and when chromatin is open, and as you're finishing up, chromatin closes. That's what that one was talking about. This figure tries to tell you that when chromatin is initially open, you have a certain set of transcription factors that are active at that time. Early in development, maternal transcription factors from the egg are the ones that are being active right now. So chromatin, as it's opening initially, certain transcription factors from the mother are the ones transcribing all those genes that need to be transcribed in development. But as the organism becomes more and more complicated, the mother's transcription factors become less important and less active, and the actual new organism, the embryo in the new organism, starts taking over transcription. Okay, that's all this one is talking about. This switch between a mother's transcription factors during development to the embryo's actual transcription factors later in development. Happens very early, as you can see, ever from the 2-cell stage onwards. Okay, other interesting things. Let's see. Okay, how to explain this. So again, he'll probably talk about it in different detail than I will. I usually try to oversimplify things so you guys get a general idea. So if he gives you more detail, you stick with the more detail that he gives. But fundamentally, what this diagram is telling you is that you see these methyl groups here. Initially, when you go from 1-cell to 2-cell stuff, a lot of these methyl groups that were originally part of your father and your mother's genomes are actually lost when a new organism is developed. So all those methyl patterns that your parents accumulate during their lives are reset when a new organism is made, when an embryo is developing. And that's because when this embryo ages and develops, it's going to accumulate its own methyl patterns. So that's all this diagram is trying to say here. All right, more stuff. I personally think, again, this is way more specific than you need. If it turns out he talks about it in detail, and if it turns out you need it for the exam, I will come back to it in detail later. But this is just saying that if you methylate certain genes, certain transcription factors are activated and other transcription factors are turned off. I'm just going to skip that. And now we get into the next bit of information. So yeah, sorry for the complicated chromatin stuff. Hopefully when we get into chapter 28 and 29, it'll make more sense then. But that's my overly simplified, confusing explanation of chromatin and development there. So, moving on to the next interesting fact, and back to nucleoside stuff, is that there are certain nucleosides, nucleoside drugs that can be used in a process called gene therapy. It turns out you can use nucleoside drugs to treat certain viral-based diseases, because you can design drugs that are specific for virus enzymes as opposed to human enzymes. So you can read this if you want more details, but bottom line of this thing is that you can design specific drugs to specifically target a virus version of an enzyme instead of a human version of an enzyme, which, believe it or not, is actually harder than it sounds. Drugs like to target multiple things, especially things that weren't intended to target. That's why certain drugs have so many different side effects. Side effects occur simply because drugs sometimes inhibit a thing they're not supposed to inhibit. Sometimes, though, we get lucky and we get really specific drugs that target exactly what they're supposed to target, as opposed to accidental side effects here. More interesting facts. This is the interesting fact section of the notes here. This one is talking about the actual drug that inhibits a viral thymidine kinase, which is the enzyme I was talking about in the previous slide. You can look at the pictures here, not necessarily important, it's just for context purposes. Again, if he wants to talk about it in specific detail in the exam, when the exam review comes around, I will talk about it in detail. But right now, this is just illustrating the concept in the previous slide. This is an interesting fact thing. So this is a performance-enhancing drug called ICAR, or AICAR, and there's the long name listed on there. The reason why this is a performance-enhancing drug is because of what it does. This drug, also a nucleoside derivative, is a mitochondrial production factor. Basically, you take this drug, and a lot of studies have already shown this, if you give this drug to mice, they end up producing more mitochondria per cell than mice who don't take this drug. And in fact, and let me see, yeah, he does have this statement in here, you can take a really lazy mouse, and this is how Hasty described, you can take a really lazy mouse, feed it ACAR, and it can run just as fast as a mouse that is trained to run all its life. So right now, there's very obvious controversy here, because I believe a couple years ago, a certain group of athletes were found taking this drug. A certain group of human athletes were found taking this drug because of its ability to increase mitochondrial production factor, because it's a mitochondrial production factor. So I actually, a long time ago, once looked this up just to see if there's a sketchy way to order this, and there is a way to order this. I do not suggest ordering anything like this, because obviously, who knows what side effects this thing has. But it's interesting to think about sometimes, right? That there may be a drug that gets you to exercise better and all that. Who knows? All right. Is that the last interesting fact? Oh dear, I'm not looking at that. Okay. Yeah, so that was the last interesting fact. So, usually, context purposes, the most common way he asks questions about all these like nucleoside derived drugs and chromatin, methylation and acetylation and all that stuff, he usually asks it in very simple ways, such as, azacitidine is a what? And the answer would be something like an epigenetic drug, or it's used to treat acute myelogenous leukemia, like simple facts like that. If he asks what ICAR or ACAR is, we just found out ACAR is a mitochondrial production factor. It is a nucleoside derivative like everything else we listed here. So this section of notes usually scares a lot of people because of how information dense it is and how many new terms they've never seen before show up. But don't panic too much. Just look at the general biological facts and biological consequences of use or benefits of using these drugs, and you'll be fine for most questions. So, finally getting past all of that, we move back to slightly more familiar things, nucleotides. Okay. So earlier we talked about nucleosides, and I emphasized the S there, and that's because there's a slightly different category of molecules called nucleotides. You see in blue here, they call it nucleoside phosphates. So what a nucleotide is simply a nucleoside with at least one phosphate attached to it. That's it. That is what a nucleotide is a nucleoside with at least one phosphate attached to it. Here's all your nucleosides that were drawn earlier with one phosphate attached to it, and you can see how the name of each one of these things changed. Adenosine monophosphate, guanosine monophosphate, cytidine monophosphate, etc., etc. They're telling you how many phosphates are there. You guys have definitely heard of the molecule adenosine triphosphate before. You now know what that looks like. Adenosine triphosphate would very clearly be an adenosine nucleoside with 3 phosphates attached to this end right here. In fact, let's see, is there a picture of it? No, of course not. Adenosine triphosphate, all it is you see this one phosphate here, imagine 2 more phosphates right next to it on the left there. That is what adenosine triphosphate would look like. So the role that these phosphates serve in terms of a nucleotide structure is it actually now gives all those nucleosides actual functional roles. So remember, nucleosides by themselves don't do anything except for adenosine. When you attach phosphates to them, they actually have biological roles. So this slide lists a couple of them. You should know the general roles listed here and the specific roles listed here in case anything is asked about. We already know that ATP is central to energy metabolism. We know that from a long time ago. And some of these other ones you probably haven't learned before, so make sure you internalize and memorize these things. When we get to chapter 30, we'll learn about GTP, CTP, and UTP. You learn about in Biochem 2, or if you're studying for the MCAT. So, moving right along. Other nucleotides, so other than the general nucleotides, which are nucleosides with phosphates attached to them, you have a category of nucleotides called cyclic nucleotides. You can kind of see the nucleotides still on here, right? You see a nitrogenous base, you see a ribose sugar, but instead of a nice looking phosphate, it's kind of wrapped around itself to hook back onto the sugar, right? If I go back to the regular nucleotides, phosphate nice and normal right here, just bonded to the nucleoside by a single bond here. But a cyclic nucleotide, instead, the phosphate wrapped back around and is bound at the 3' end here. Certain molecules, such as, is it listed here? Yeah, cyclic AMP and cyclic GMP serve signaling functions. In other classes, you learn about a cascade called the G protein cascade or the G protein pathway. You won't need to know that for this class, but the G protein pathway requires cyclic AMP because it is a signaling molecule, so it communicates to other parts of the cell to do certain activities. So that's the general activity of cyclic nucleotides. So after nucleosides and after nucleotides, now if we start hooking these nucleotides together, we finally get to stuff that is important: DNA and RNA. DNA and RNA are polynucleotides. They are polymers of nucleotides. Let's look for a nice picture of one. Here's a polymer of nucleotides. All these nucleotides with phosphates in between each one of these nucleosides here. The one on the right represents DNA. The one on the left is an example of RNA. These are polynucleotides, polymers of nucleotides. So let me check. I'm going to make a very important distinction here in terms of numbers and directions. And I will probably repeat this again when we get to chapter 28 because it's an annoyingly confusing thing here. Okay. So I'm going to write out a DNA sequence. I'm going to label it this way, and I'll explain why. So, a lot of times when DNA is written out or drawn out, they label two specific components of it. An end of it called a 5' end, and an end of it called a 3' end. There's a specific reason why they draw it in this direction here, is because this is the direction that we read DNA in. We read DNA in a 5' to 3' direction. So this, whenever a DNA sequence is written out, very often they write this number here on the left, and they write this number here on the right to indicate which end is the quote-unquote front end of DNA, which one is the back end of DNA. The reason why we read DNA in this direction is because if you carry this direction all the way down to the translation step of the central dogma, if you take DNA, you transcribe it, and then you translate that mRNA into protein, this direction corresponds to the N-terminus of an amino acid sequence. And you can probably rightly guess that the 3' end represents the C-terminus of an amino acid sequence, which we learned way back in exam 1, that the N-terminus was the front end of a protein, and the C-terminus was the back end of a protein. So that's why we read DNA in a 5' to 3' direction. That when we read it left to right, the left side is always 5' and the right side is always 3'. Okay? It's very important to understand this direction here. The reason why I make a big deal of it is because when we get to transcription and replication even, so we're going to see this in chapter 28, I'm just going to briefly talk about this one component here, is that if I have a template strand of DNA, if I want to replicate DNA, you'll learn later that it does so by the two strands of DNA end up separating into single strands of DNA. And to replicate it, you have enzymes that read a template strand of DNA and just copies that template with a corresponding strand of DNA. Turns out enzymes read template DNA the other way around, in a 3' to 5' direction. That the enzymes responsible for replication and transcription read DNA the other way around. Okay, they read it in a 3' to 5' direction. The reason why is, I'll draw enzyme right here. It's going to read the template strand right to left, right? Because that's the 3' end and that's the 5' end. It's going to read it in this direction, because it's going to make a new strand of DNA going right to left. And it's going to make the new strand of DNA 5' to 3'. So let me write that out, because it sounds backwards, right? Enzymes read template DNA in a 3' to 5' direction because it makes new DNA in a 5' to 3' direction. You might remember from earlier biology courses, they use a phrase, what is it called, anti-parallel to describe DNA. If you don't know what that means that the two strands of DNA are oriented in an opposite direction. One strand of DNA, which is your old template strand of DNA, is drawn 5' to 3'. Left to right happens to be 5' to 3' in this illustration. The other strand of DNA, therefore, has to be the other way around. The other strand of DNA, in an illustration of double-stranded DNA, the right side has to be 5' and the left side has to be, the rights have to be 5' and the left side has to be 3'. The only way this is able to happen is because enzymes, when they replicate DNA, they do so by reading the template strand as I have drawn here in a 3' to 5' direction and copying the template strand into new DNA, making it in a 5' to 3' direction. All right, so let's review the 3 different directions we have, because I can't tell you how many times people get this confused. If we, as people, are given a sequence of DNA that we're just reading on an exam or reading on a piece of paper, and they don't happen to label the ends of it, we can assume that the left side is 5' and the right side is 3' if we are reading DNA, just on an exam or in a paper, that the left side is 5' and the right side is 3'. Biologically speaking, so that's us up here just reading it. Biologically speaking, if we're given a strand of DNA, if an enzyme is given a strand of DNA, it's going to read it in a 3' to 5' direction so that it can make new DNA in the correct direction, 5' to 3' because of this anti-parallel arrangement. And I'll address this again when we get to chapter 28, but I mentioned this here because on the PowerPoint slide, they talk about directions of DNA. And this is the reason why we have these directions of DNA, is this orientation mechanism here. Okay. One more reason why there's this orientation stuff. Let's see if the picture quality is good enough. Not really. Okay. And then I will draw this part then because the picture sucks. One last reason why we have DNA written in that direction, or what the direction means. I'll check the clock. Yeah, this will be the last thing we'll talk about today. Alright, so this is a overly simplified picture of a polynucleotide. Alright, I've drawn two nucleotide components of this polynucleotide. Each one of these is a nucleotide. We can tell because there's some nitrogenous base attached here, there's a ribose attached here, and there's a phosphate attached here. And notice it's connected in this orientation here. Where the numbers come from, where 5' and 3' comes from, is what number on the sugar that the phosphates are attached to. Or if you go back to exam 2, you'll remember how to number carbons on a sugar. The one that the nitrogenous base is attached to is carbon number 1. This one's carbon 2, this one's carbon 3, this one's carbon 4, and this one is carbon 5. The reason why we write DNA in a direction of 5' and 3' and where the numbers come from is because one of the phosphates is attached to carbon 5, and the other phosphate is attached to carbon 3. That's where it comes from, okay? Note specifically we use 5' to 3'. It isn't usually explained why, so I'll give you the reason why. The reason why we attach this prime thing here is because that is specifically to describe position on the sugar. If you ever see it, you won't see it in this class, but if you ever see numbers written like regarding a nucleotide like without prime numbers, they're referring to carbons on the nitrogenous base. That's a subtle difference, but that's why for sugars, for the nucleotide part, we use primes, is because we're describing the phosphates and the sugars down here. But if you see in future context any numbers written without primes, they're talking about any position, some given position on the nitrogenous base itself. Okay, so that's the kind of annoying description of direction on here. We'll continue with nucleotides next time, and that's it for today. So I'll see you guys after spring break.